Thank you everyone for coming in. First of all, thanks to Richard and the club. This is really a great place. And I thought I would just take a minute and introduce Maaz and myself. Maaz and I, we've known each other since 2013, the first semester, and it was a coincidental meeting. We've been very good friends. We've been in touch for almost 8 years, and it's really a pleasure to be doing this. And I hope that you would enjoy this. Just a disclaimer, I'm a medical student and Maaz is doing his PhD, so hopefully it will give you a complementary combination. I'll pass it on to Maaz for the introduction. Thank you.
Thank you, Saman, and hello everyone. It's good to be back. It's good to kind of share our experiences. So I'm going to be pulling up my slides. Like Saman said, I'm a PhD student currently at NYU School of Medicine. I'm in my second year, and I was at Berkeley from 2013 to 2017. I was really involved in the stem cell community at Berkeley and even afterwards. So I'm going to be talking about the importance of plasticity. So this is a concept you might have heard in the context of cells, but I'm going to, make an inference and kind of talk about how it is important in life as well in my entire journey as a student doing research. So to briefly touch upon like the agenda of my talk, I'm going to be going over the anatomy of a crisis, creating value, what does it take to be successful, and then my own personal journey. I'm going to leave you with something a bit more experimental. So, for those of you who like more, technical stuff, the kind of research that I presented that prepared me for grad school. And then we can have a 5-minute Q&A, and then I'll hand over to Saman, who's going to share some of his stuff with us. So the reason why I'm talking about a crisis is because a lot of students at Berkeley, including myself, at some point go through a situation which is unexpected and a situation which kind of triggers a sense of urgency. And you sometimes are lost, you want to do something about it, but there's, a very limited amount of things that you can think of during that time. And I've seen that a lot of successful people have this ability to not lose control during these situations. And this is one of the books that I read a while back when I was at Berkeley, Man's Search for Meaning. This is by Viktor Frankl, who's a scientist, a Holocaust survivor, and just an incredible human being. And the way he describes this was between stimulus and response, there's a space. In that space is our power to choose our response. In our response lies our growth and our freedom. And that's something that really resonates with me because I've had situations where I didn't get the opportunities that I wanted, and just thinking about like what I could do during that time has really made the difference. And I'm going to bring up this diagram again because I find that this diagram has evolved over many years. So in my first slide, you might have seen that there wasn't as many arrows from one point to another. And for those of you who don't know about this diagram, this is a cell state diagram, which is kind of the backbone of stem cell science. You have a stem cell, which kind of is able to differentiate into different cell types. And, you kind of go into these different valleys of cell states. What's interesting now, like in 2021, is that this diagram has completely changed. Now we've realized that we can go from one side to another because of plasticity, because of being dynamic. And this is an ability that is important in life as well, because you might be in a certain situation where you might think that you cannot go back or you cannot do something about the situation. I can assure you that that's not the case, because at the molecular level, life is built in such a way where you can always reverse trajectories and always try new things. So I always thought of this as a somewhat of an inspiration to be able to do the things that I wanted to do. And more recently, I was at this talk from one of the board of directors of Moderna, and they brought up this really good point about what does it take to be successful. A lot of people have different views about this, but the one that I kind of believe in is you have to be different, but you also have to do it exceedingly well. You can't just be different and not be better than others. So, and the reason this is important is when a student was applying to grad school or medical school, you have to stand out. And there's a lot of people who are doing like important research, but it's not enough that you were able to just like show what you've done. You have to kind of stand out from the pack. and show what you've, like the work that you've done, why is it important? And this is something that I realized like pretty late during my career, because I was not really focused on this when I was at Berkeley. I'm going to mention this when I talk more about my personal journey. So it's a long time looking at this picture again. This was a conference that Saman and I were part of, stem cell conference, a couple of years back. I think it was 2015. So back in Berkeley, I was looking for research opportunities, but I was kind of lost, honestly. And Saman and I, we decided we were going to join, the Stem Cell Society and kind of learn about stem cells, because that's something that was quite interesting at that time. And we were always thinking about how do we get into research? And it was just a very difficult time at that, and we were, trying to always come up with new ideas. And at that time, the only thing we were able to do was volunteer or talk to professors, stuff like this. And this was a kind of a crisis situation for us, because we didn't have the things that I mentioned on the left side, which is cognitive complexity, critical thinking, openness to new ideas, creativity, risk tolerance, all of this stuff. So how do we get to this point? I mean, we have to do something unconventional. We have to come up with new ideas and get to the place that we want to. So at that moment in time, even though I was, reading about stem cells, kind of doing the work, kind of reading about new papers and techniques, I wasn't able to physically do them myself. So I thought, if I'm not going to get into a lab at Berkeley, why not kind of think broad and maybe apply to labs all over the globe, look for opportunities globally. So, and that was a moment in time where I actually felt like I could do something about this crisis, and I could break the spell, find an opportunity. So, and that brings me to my journey in Japan. So I actually took a year off and I traveled to Japan. It's the hub of stem cell research. And I was working with Dr. Yoshiki Sawa and his accomplice, Dr. Masuda Sensei. You can see him in one of the pictures over here. And it was just an incredible experience, kind of making friends, doing research in a completely new environment, and just breaking barriers, scientific barriers, cultural barriers. There's a lot of things that we don't think of that goes into good science. And one of them is being able to talk openly with people around you. And that's incredibly difficult and challenging in a new place. But that's what this journey gave me. It gave me a lot of confidence, allowed me to do this kind of research, in a very short time, but also something that I would be able to use in the future. So moving on, I think I mentioned in one of my, actually, I might not have talked about this, but when I was doing research in Japan, I was really tired and kind of frustrated about my results. My experimental results were in 2 dimensions, and I wasn't getting any good data from my experiments. So 2 dimensions means that you just culture cells on a plate, and you were able to do some assays and experiments, and sometimes that's not reproducible. So when I came to Berkeley, I had this renewed vigor that I wanted to do something with 3-dimensional systems, so I could, make more of an impact. So I decided to come up with a team of Berkeley students, and we decided to make this initiative called Timorous. And we got this funding from NSF, and we did this whole I-Core program. We didn't incorporate eventually, but it was just a really good experience, kind of thinking about how to build stuff from scratch and to solve a problem. So this was an opportunity that allowed us to talk to people at Genentech, Pfizer, Lilly, and kind of understand the whole drug screening landscape. So now I want to talk about very briefly about some of my work, my research work that I did in Dr. Shinya Yamanaka's lab. So I wanted to also use this slide to explain the whole journey from Berkeley to Japan and back and to Dr. Yamanaka's lab. I remember when I was sitting with Saman organizing this conference in 2015, we decided to email Dr. Yamanaka, who was at that time very famous because of his work. And Saman and I, we sent this email, and Saman made a bet with me. He's like, you're never going to get a reply, even in the next 6 months. So there's no point even sending a follow-up email. So we kind of gave up on our plans, and we just decided to kind of call it a night and look for other speakers. But here I was like back in 2017, and when I came back from Japan and did this like research in Berkeley, I applied to Dr. Yamanaka's lab at UCSF, and thankfully I got in. And it was just this culmination of all these different ideas over the years and lessons that I learned that allowed me to get here. And the reason why I wanted to talk about this stuff is just to give you guys a brief idea of like what it means to present some of the work, as a presentation, or just a verbal talk at your grad school interviews, or even in your essays. So I'm going to briefly mention this project. It's about NAT1, which is a translation initiation factor. And before I talk about NAT1, I want to briefly explain like what are stem cells and why they're important. So I'm sure you guys have seen this before. This is something that is taught like at every stem cell meeting, that they have two qualities. The stem cells can self-renew, or they can differentiate into the 3 germ layers, which can be used for organ transplantation or studying like organismal development. But this part, self-renewal aspect, is actually quite interesting. And this is what Dr. Yamanaka wanted to study. So we wanted to think about like what was going on. And this was one of the experiments that was performed by Kazu in the lab in 2020. And this experiment showed that this protein called NAT1 was required for iPS self-renewal. So if you look at the figures, the top panel, you'll see that there's wild type colonies. iPS cells actually form colonies that are quite tightly packed. And then over time, when you remove NAT1, the colonies become really small. They actually lose OCT4 expression, and they become differentiated. So this is a phenotype, and it's a very strong phenotype of loss of self-renewal if you remove one factor. So we were really interested in what was going on, and we wanted to further explore how we could, use this phenotype to discover something biological, something mechanistic. So the big question was, how does NAT1 regulate self-renewal? In order to answer this question, it's important to know what it is, right? So we wanted to deep dive, but also at the same time look for literature and other things that had been published at that time. And one of the things that we noticed was that it's very similar, NAT1 is very similar to this protein called EIF4G1. So if you look at the diagram on the left side, this is a diagram of an mRNA that's being translated. An mRNA has a poly A tail and a methylguanine cap, and that's required for this mRNA to be translated. So there's this protein called, so the main point here is that there's a cap which needs to be recognized by EIF4E, which is a cap binding protein. And then there's a poly A binding protein which binds the poly A and circularizes the mRNA. So this is something that happens with almost every mRNA in the cell, or most mRNAs in the cell. And as you can see, this EIF4G1 is kind of like a scaffolding protein. It's bringing everything together. And it brings, the EIF3 machinery, and then there's a ribosome that sits on the start codon and translates into protein. But there's this like obscure protein, which is similar to 2/3 of EIF4G1, doesn't have the ability to bind the cap, doesn't have the ability to bind the poly A, but yet is critical for the cell. So everyone was like picking their brains at that time. It was like, what is this protein doing? Like, how is it able to like bind mRNA and do what it's doing? So this was a big question. It's something that people are still figuring it out. Like, we don't have a clear idea of how this works. But there are some theories, like, some people think that there's a EIF3D, which is one of the subunits of EIF3, which binds the cap and then allows NAT1 to translate mRNAs. But I just wanted to bring this up because this is something that is still being figured out. It's a big question. So the question, one of the experiments that the lab performed was doing a affinity purification mass spec. So this is a technique where you tag your protein of interest with a 3x flag, and then using this 3x flag, you pull down and look at the interacting partners. So for every good experiment, you need to have a control. and our control was EIF4G1. This one, just to remind everyone, has both the ability to bind the cap and the poly-A. So it's the canonical translation factor, whereas NAT1 is this smaller version of EIF4G1, doesn't have the ability to bind the cap or the poly-A. And we wanted to look at like what are the factors that are enriched in either condition. And what we found interesting was that there's a set of proteins called PRRC2 proteins, which are really highly enriched with NAT1, but not very highly enriched for 4G1. And my project was to figure out like what these proteins are doing. So I had this data in front of me and I was thinking like, what are the next steps? Like, how should I explore this problem? Yeah, thank you for having me and Saman, and please let me know if you have any questions.
Yeah, that's absolutely insane. I mean, we literally go over the Yamanaka paper like so many times in like bioengineering curriculum for like the people in the cell and tissue engineering concentration. So it's amazing that to see that you were actually able to get into like I think a Nobel Laureate's lab, right, for iPSCs. So how did it feel to be working under the person that literally invented iPSCs?
Yeah, no, thank you. I think this is a great question. I was very intimidated at the start because Dr. Yamanaka actually lives in Japan, so he used to fly every month just to kind of meet with the lab, and you have to have an in-person meeting with him, so you have to present work. So it's very intimidating. But I've realized that people who've actually made a lot of scientific discoveries have a very different way of thinking, approaching problems. So it was a very nice experience kind of learning from him, how he thinks, how he approaches problems. And overall, it was very, very nice being with him for about 3 years. So overall, it's not different from any other lab, I would say, but it's just nice to be able to see how these people think.
I don't want to take up more time with my questions, but I'm actually really intrigued by the project you started at UC Berkeley. I forgot the acronym for it, but it was for like reducing the instance of like false positives and false negatives in a device. And I thought that was super cool. Just curious, what kind of support did you receive from the university, or support did you seek out when you were trying to get that device off the ground? Like in terms of like funding, or maybe like advice on approval, etc.
That's a great question, Richard. I think Berkeley has a lot of resources. I was very interested in bringing the device to the market, but we made a lot of mistakes initially thinking about, this is how we're going to get into the market. And in terms of funding, very easy to actually get funding from different sources. We were able to apply for funding from different accelerators and startup like incubators at Berkeley. We only got like substantial funding from NSF, which was around $50,000 like to begin with, to do like customer discovery interviews. And then if we had followed up, we could have applied for the SBIR grants, which go up north of like 250K. So it's really dependent on like where you see the project going. But I think the mistake that I made in this project was thinking solely about the device and not thinking about the whole technology as a platform, because once the device is out, you have to start thinking about the future, about like what's going to be there next. And in my 100 interviews with like directors of drug discovery, I found that people are actually not thinking about drug screening the way I was. So it's important to really scope the market before you actually build something. And that's kind of the advice that I have, even though I know I'm going on a tangent.
In regards to work under Dr. Yamanaka, what would you say was the most like out of box problem solution that like for issue that you thought was impossible to solve originally?
Yeah, that's a really good question. So I would preface by saying that 90% of the experiments that I tried in the lab were failures. There's only a few experiments that kind of worked. One of the really challenging experiments was ribosome profiling. So this is a technique that was actually developed in Berkeley in the Ingolia lab. And it involves like footprinting ribosomes on mRNA and seeing like the presence of ribosomes and that kind of tells us about like how well an mRNA is being translated. So I was trying to do this technique, and it's a 7-day protocol, and it's sometimes like you really think about like which steps are important, and you don't want to make a mistake. But at the end of the day, it's all about like thinking about the big picture, because if you get like lost into the details of the experiment, you sometimes like don't understand why you're doing it. You lose motivation, you make mistakes. So for me, it was really important to kind of take a step back every day, think about the bigger picture, and then speak with experts, obviously. So I was in touch with the Ingolia lab, Dr. Jamie Cade, to kind of like always understand like the issues that I was dealing with. And eventually, after many runs, I got it to work. And it's really about like grit and perseverance, I would say. So I'm not a straight answer to your question, but I think it's a lot of different angles that you have to think about.
Yeah, okay. So thank you so much, Maaz, for your insight. For the sake of time, I think we'll move into Saman's talk.
Can you hear me now? And can you see my screen?
Yes, we can see your screen.
Okay. So what I will do, and it's a little bit hard to see everyone, so I'm going to try to make this work. This is going to be a little bit different. Let me also set the timer up because I tend to go over time. The focus of this is more a reflection than my research. I mean, there is content there and I'll present it. That's not a problem. But I think what people want to hear more is perspective of people that have been at Berkeley and are doing something different, and perhaps some sense of, what is it like and what transformation is like. I really appreciate some of the themes that Maaz has brought up in his talk, and I'm probably going to use some of that narrative to continue forward. I will be happy to entertain questions during the presentation as opposed to at the very end, because I think it's more interactive. So if you have questions, I can't see the chat, but just unmute yourself, okay? And I will pause before every slide so that you can do that. So as you can probably tell, I'm a little bit unfocused. I'm interested in a lot of different things, and that's always been a theme for me. So it's a bit unusual perhaps as to, by the way, you can add me on different platforms. I'm happy to provide mentorship to people because I was a beneficiary at Berkeley, having strong mentors and friends. So if you're applying to medical school, if you have questions about medical school, if you have questions about securing labs, anything, just message me and say you attended the talk. It will be a pleasure. Okay. So about myself. So this is the logo of the, can you see my mouse? Probably not. Can you see my mouse? No. Yes. No, I guess not. This is the logo of the club back when it was SSCR, and Maaz and I actually joined in our first semester. This was Maaz's suggestion to join a club, and I was reluctant initially because with Chem 1A and all the other classes, it didn't seem like a wise idea because, it's quite a grilling experience. But I'm very happy to have done that, and I'm very happy that we sort of continue this. Some trivial facts, because I mean, it's really nice that there's an alumni event and we're having people that have done other work. I think sharing your failures also is important. So when I had a little bit of that here and there, Maaz and I both attempted to run for presidents and we both lost. So that's just a small thing. So just something to note. There's a lot of mini failures, and I mean even the concept of failure itself is poorly defined. When we think about failure, is it entirely outcome centered, or is it something that the unattainability of achieving goals, or really what exactly is failure? Doesn't have to be professional, that would be personal. So I'm Persian, if that helps. That's my name. My name means organization, although we'll have to see how organized the slides will be. So there'll be a commentary of that. I did my high school in BC, Vancouver. With my whole concept of Berkeley, I remember having this chat with Maaz, and I told him, I really want to do these different things. So I did a MCB degree in Persian literature. I did a minor in chemistry and music. And the chemistry was quite brutal because I decided sort of midway through my third year, so rather late, that, I really want to do this. And the concept that I'm going to really highlight here, again, this is a probably I can't say ill-advised because there's merit to it, but it's an uncommon advice you're going to hear from someone that is in medical school. And the idea is that the best time to take risks is in one's youth, and the best time to take risks is to know what is worth it and what is not, because the gains can be quite significant. I was involved in a bunch of laboratories at MCB labs at Berkeley. No publications there, though, I have to admit. I was part of some really good work with researchers and learned a lot from them. I came back to Canada, and the story for me is a little bit interesting because I had initially intended to stay in the States and apply for the MD PhD, but I'm an international student. In fact, both Maaz and I are international students. It's another factoid about us. And so I realized that the funding isn't really available to international students, and the tuition is quite insane. So I made the wise decision to come back to Canada, where I was a citizen. And I moved to Toronto initially for about 2 years. And then I moved to Quebec, Montreal, where I'm a medical student now. I'm a second year McGill. So there's a little bit of that. Not too much to talk about here. Probably the more important things are here. So when we think about the developments of a mind, I mean, whatever way you want to think of it, whether you want to reduce it down to individual parts, where you want to think of it as more Gestalt idea, it's an assessment, right? And I like to think of it as a web, as a spider web. We have all of these different nodes that are sort of interacting with one another, and there's interrelationships between them. So one of the things that you probably will find at Berkeley, I'm not sure if is it still there, the letters in science breadth requirements, like you have to take 7 classes, different partners, is still going on like that. That tradition really extends back several hundreds of years, and that's probably why the degree in MCB is still a Bachelor of Arts, not a Bachelor of Science, which was something that I was always a pet peeve of. But really the idea is that the more you develop your senses in different disciplines, you can find connections. And this is certainly true for, I mean, Maaz is a great scientist doing really great work, and people who switch from different areas, like if you have a more genetic focused background and you switch to more cell biochem based, there's a lot of creativity there. So one of the things I really want to highlight is to really expand your horizons in ways that you can challenge yourself and that you can find connections. And that's really very important as we move forward. Okay, I'm going to stop. It's been 5 minutes. I've been keeping track of time. Any questions, anything so far? Are we good? Should I continue? I guess I'll continue. Okay, so my interests are, again, very diverse, and I think that's probably the part that I want to highlight. One of my obsessions, and it still is an obsession, is books, and I've always loved collecting them, reading them, not reading them, smelling them. And the reason for that is that when we think about thinking as a principle, right, thinking is a way that we get to regulate and manipulate information. And that's really important, especially with people that have aspirations to do basic science research or people that want to go to medical school. And the idea here really is that how do you challenge your biases? And it's a very difficult question you have to think about, because in order to challenge your biases, all of us live with a set of principles. We think a particular way about ourselves, the way that we have an identity. We think very specific things about how we interact with other people. And sometimes we need to challenge it at a very axiomatic level, at a very fundamental level. Like, why do I believe in what I do? Why do I want to pursue what I do? Maybe some of you are sort of in between. Should I go to medical school? Should I go to graduate school? Should I go to education? What have you, right? And I have some lessons in there too. And part of the good life is maybe addressing some of those. So this is back when I did it.
Saman,
go on.
I just wanted to address that there's currently a question in the chat about your
Would you mind
minor
reading
in music.
Okay.
Yeah.
Sure. What was the question?
Let's see.
I can't read the question.
How would you suggest balancing academics with research and other passions? I saw that you minored in music, and that's something I'm planning on doing too, from Clara.
Thank you very much, Claire, for asking this question. Yes, I did minor in music. The most important thing to do is to give yourself a certain buffer zone, and that's what I did not do. If you want to minor in music, I'm assuming, is it performance? Because I mean, Berkeley has a musicology department, but there are pianos available. The best way that you can do something is to plan things out, and we're going to get to that slide. And the best way to plan things out are not all the things that will go right, is to plan for all the things that will go wrong, right? And having a contingency plan that allows you to think about how you're going to navigate through this. Maaz talked about crisis. One of the things that is important, studies in social psychology show this, is that if you have some degree of mental preparation for something, even if your coping mechanism is ineffective and is not going to work, you're not going to be in the shocking zone. Now, with a minor in music, I think the best part that you can do is to spread it apart. I'm not sure what year Clara is in. If you could clarify that, I could maybe help. The way that I did it was I sort of spread it within 1 or 2 classes within the semester, with the added caveat that if any of you have taken MCBC 108 by a physical chemistry professor Kurian, I remember this very well because I had a music class and the same class overlapping at the same time, and I couldn't change it because I needed the degree. So what I did was I went to the music class and I messaged Maaz whenever, because there's polls on that class, right? iClickers. He's like, there's a question. I'm trying to sort of figure it out within the 2 minutes or so. So don't do that, right? It sort of worked out, but don't do that. So I'm happy to, of course, address more details that Clara has. I don't know if I answered the question completely, but I'm happy to go into more detail later, and you can contact me. So this research is back from 2016. This is the SURF. I did a research that involved literary theory and philosophy. So I looked at historical consciousness, and so this was an interesting part. Sorry, a question? I see this. Oh, okay. I thought I heard something. So it was a very fun experience, and I really enjoyed that. So again, why would you have, or maybe a question for me is, well, why would you be interested in neurobiology, music, philosophy, and medicine, right? If you can think about the world as categories and concepts, you're going to have an easier time navigating your way through, right? So on a molecular level, neurobiology provides us with a very clear, reductive understanding of behavior, of learning, memory, what have you, right? Philosophy is thinking about thinking itself. So it's a more abstract way of looking at something that's reductive, right? And music is different in the sense because music sort of transcends beyond the body and beyond the mind. It's something that encompasses it all together. And medicine, I probably should maybe mention this too. A lot of times when I talk to students and people that are interested in medicine, they share a very early interest in medicine. I was not the case. In fact, my parents are both physicians, and they told me not to go to medical school. And of course, being a great child, I did exactly the opposite of what I was told. I applied for medical school. And I think that it's important that when you form your identity, you try to ask yourself, what is it that you're interested in? And it's important to recognize that there's a sense of development to what we do. Maaz and I are still students who will continue to be students. We're just in a different phase of our life. It's not like we are the know-all, end-all just because we're alumni. But that development and progression is a gradual step. Some of it you can regulate and control. How do you do it? You find a Nobel Laureate, you work in his lab, you try things, right? Some of it, you try things and it doesn't work, and again, you think about what you can do. But so long as you have some sort of contingency and you have good mentors, you're bound to succeed. It's guaranteed. That I can assure you. Okay, let's continue moving forward with the slides. Okay, so one of the things that I did, I did do some hospital experience, but again, something uncommon was that I spent the majority of my time, especially after graduation, because I was still in the States doing research at UCSF, was at the Friends of the Berkeley Public Library. And this was a bookstore. It's between Durant and Channing. I recommend you to go there. I'm not trying to advertise for it, but they have great books. And what I love about this experience and volunteering there was that I came across so many different things that I did not know about. Areas like anthropology, like linguistic philosophy, or even chemistry stuff. And what that encouraged me to do was to try it, to learn something. And I firmly believe that if anyone reads one page of a book, that was time well spent, even if that's the only page you read. So the idea of encouraging you to try things, because really trying to get into medical school in North America, Canada, the States is extremely competitive. There's no denial of that. And we can maybe talk about strategies or things you have questions in particular at the end. But it's important to give yourself that 10, 15 minutes a day to satisfy your curiosity outside of things, right? Not everything has to be related to research or to medicine, right? Maaz has an interest in cricket, for example, or he has interest in, for example, clones and collections. He didn't talk about that. But it's important that you maintain things that you love, because you live your life for yourself, and you cannot sacrifice everything for your professional pursuits. And I did that. I did that for some time, right? So this is something that if I went back in time, I would have loved to change, right? So give yourself that liberty, even if it's a few minutes, and it's essential. So that's another point. I'm trying to be making this a little more reflective narrative, so hopefully that's okay. Now, takeaways about being at Berkeley, and I'm not sure what year you're in, but some of these concepts I think are very important. The first is collaborative learning. If you're enrolled in a class, it's new for you, you don't know any friends, reach out to people, sit in the front row, always sit in the front row. I have issues with my eyes, so I have to sit in the front row, but aside from that, it's really important that you just start conversation with people. Because I can think back, and if I didn't start that conversation with Maaz, I would have never known him. And that's a pretty crazy idea to think that someone I've known for 8 years, I would have never really had a chance to know just because of that instance, right? There was, because he passed away recently, there was a famous psychologist at Stanford, actually Canadian, Albert Bandura, and he talks about fortuitous events, and that a lot of times when we are doing things, chance plays a big role in the decisions we make. But those decisions can be very everlasting. So that's one thing. Try to work in groups, study with groups. I know the classes are curved to a C- for almost everything, but you can find people that you can work with and you can engage with and you can learn from, because when you explain something to someone else, you gain a mastery of that, and that is central. So that's one area. The second is prospective planning and strategic thinking. I've sort of addressed this a little bit earlier, knowing that things can go wrong and how you would circumvent that. Having a sort of a logarithmic plan. For example, these are things I like to do the next semester. So that's obvious. It's sort of, you have to do it because CalCentral, what is it called? CalCentral still, I think? You have deadlines, all that. That's short term. What's long term? Long term is, well, these are the other areas I would like to explore, because maybe Alexei Filipenko is at Berkeley and I like to take a class in astrophysics, and that's interesting. So that's academically based. Prospective planning also means giving yourself time to know yourself. And that means just silence of the night and just thinking about who you are, right? So you think about who you are, and that helps you in forming your goals and aspirations, and also your personal relationships, which are extremely important with your friends, loved ones, significant others. Very, very important. Okay, the next idea is networking as a grassroots concept. Having left Berkeley, and I think I can say this both for myself and Maaz, the intensity and the intellectual rigor at Berkeley is outstanding. I mean, there are some fantastic schools out there, in Berkeley, certainly one of them, but the quality and the caliber that you notice in Berkeley is so significant because it sets standards for you. So for example, I put myself in very demanding coursework, and some I did well and some I didn't, but the intensity of that curiosity, or the standard of excellence, has remained with me to strive for bigger and better and to constantly want to improve. It's an intrinsic thing, but it came from Berkeley, and I think it's extremely valuable. So remember that. Get to know people outside of your discipline as well. That's also very important, because you get to really learn about narratives of people. I sound like an old person saying all of this, but I think these are important things to keep in mind. Know when and how to take risks, and you must take risks. Randy Shekman said this himself. He said, if you're young and you don't take risks, you're never going to take risks. I mean, they should be calculated risks, of course, maybe jumping off, I don't know, the Golden Gates, or doing something like that. Let's do something that you think has some value and that you're uncertain about some aspect of the outcome. Do not ignore things or sort of circumvent them just because of the fact that there's an element of uncertainty. So that's something to keep in mind. Okay. Optimize stability. Maaz talks about this, somewhat of an imposter syndrome perhaps, but there's a constant pressure, right? Because everyone is doing everything, right? But you need to have some sense of stability. I mean, I remember staying up the most. It was terrible. I'm not proud of this, but I stayed up for 3 nights in a row because there was just so much going on. I had all these classes. Granted, taking 8 classes in a semester is not a good idea, but optimize your stability, because you need some moment of mental clarity for yourself. These, I think, are 5 big takeaways as an undergrad. It applies to other things. So I will keep that. I don't know why I added this, perhaps because to get a sense of what sort of things that I do. Now, as you've probably heard me speak, it's not a very typical medical student telling you about all this information, but if you were to think about things that you want to have or you want to develop, find positions that you can grow in. If you're part of this club, let's say you're VP something or you're representative or what have you, stay with the club and grow in it, right? That's a very important trajectory. That's the basic concept. try to also find something that provides service to others. For me, the biggest area was suicide prevention and mental health, specifically suicide prevention, because it is a topic that I care a lot about. And actually I did some research in music and medicine on that topic. I'll maybe mention that later. But becoming an advocate for other people, even if it's in one domain, allows you to gain a sense of humility, because your time is being used for serving another person. And that itself gives you a sense of gratification, and it's valuable intrinsically. So there's a bit of that. I did some teaching. As you can probably tell, I like to talk. So teaching is a good way to do that. People have to listen to you for the most part. I did something that wasn't part of the standard curriculum. I was part of the Berkeley Philosophy Forum, which I think still exists, and we have some outstanding speakers that come and speak. So there was that. I did a lot of debate too. So I was part of UC Berkeley Alumni. So again, some list of things. Again, I didn't do this because I looked at a rubric. I didn't do this because I thought, well, what would schools want? But I had some sense of balance too. So these are some things to keep in mind. This is also a very beautiful moment. We had it wasn't unfortunately part of the stem cell club, but it was the MCB USA. And similar to what Maaz says, I guess we both have a Nobel connection in some capacity. We invited Jennifer Doudna, but when I say we invited her, I sent her an email a year in advance, and I was like, she's not going to open the email. And she replied the next day saying she's going to come. So it's good to know when you should be intimidated, but going out of that can lead to some wonderful experiences. And in fact, her talk is actually on YouTube if you're ever interested in reading it, learning about it. This is before she won the Nobel Prize. So yeah. Okay, this slide, I love this. This was in 2015. I was sitting in Professor Courion's class, and I thought about the Berkeley experience in a way. And I took this photo, and I had to go back on my Instagram to find this. It says, "Eat well, stay fit, die anyway." So is it really an accurate representation of the Berkeley experience? Is it worth it? I don't know, maybe it's exaggerated, it's hyped up a bit. But the definitive answer is yes. Because it's not so much, I mean, certainly the prestige of the university is wonderful, and that's something that stays with you as a badge of honor forever. But if you are in a stage, and usually people start around 17, 18 or later, and an earlier stage, your brain plasticity governs much of your future behavior. So the way that you are and being in that setting rewires you. And you will see that impact later on. We graduated, Maaz and I graduated in 2017, but some of that has stayed with me, right? So let me also talk a little bit about this. As you can tell, I don't like to read from slides, but I think the important points are there. There's pleasure in pain. And the reason I say that is that you really need to put yourself in positions that are uncomfortable, not because of resilience building and all of that, but because it is a position of privilege to be in, to have the option to go in pain and then opt out of it. A lot of people don't have that. It's default for them, right? So that's something to keep in mind. Challenge yourself to the core. I really believe this. I really believe that the more that you challenge, because here's why. If you think about development, there's a really nice, it's an outdated idea, but this Soviet educational theorist, Lev Vygotsky, he talks about this, it's probably on the MCAT, I don't know. Zone of proximal development. The idea is, and it's true for anything, there's a certain limit of what you can do. It doesn't have to be performance-based or intellectual. There's a certain level of what you can do. That breakthrough, that threshold, that blip, think of an action potential maybe, that is a new level. So that opens your window a little bit, and then a little bit, and a little bit more. These things summate over time, and you will see the difference. And the first step is challenging yourself at the core, right? Berkeley, again, it's a privilege to be there. It's really, you can just literally go to all of these seminars. There's postdocs coming in, there's professors coming in left and right, and you don't really get that quality. Most places, I mean, with COVID, everything's online. I'm not sure if your classes are going to be in person, I think next semester or something like that. But that's really great because you can spend some of your spare time attending some of these without pressure. And I've attended seminars that I understood nothing. And I loved it because I'm like, wow, look at all the things I do not know, right? So that is also a nice experience. Maybe you might want to do that, but something to keep in mind. Develop your sense of style, not just for speaking, writing, or thinking, but the way you like to approach things. You need to have some sense of autonomy in order to thrive. And You can always ask people about corrections or adjustments, but you must maintain that autonomy for yourself. It's very, very important. Again, competition is good because, I mean, the root of the word competition, competere, means to work together, I think, from Latin. So again, there's a lot of that as well. And external and internal push, it gives you a flavor of dynamicism. Being dynamic is important. So the Berkeley experience is worth it. And the lessons that you learn will stay with you for the rest of your life, which I think is a pretty significant statement to say. Okay, I don't know why I put this there, because I thought that we're going to talk about, I mean, let me look at the timer. Wow, 23 minutes already. I thought it was 5 minutes, but we have a tendency to do that. Okay, so I'll make this a little bit more quick. Some of the work I've done before. Maybe I'll just talk about this one. So I'm interested in mental illness and creative musical genius. And I did this sort of project here at McGill and the presentation. And it sort of worked out really nicely. And I contacted some professors and worked with them. And initially I thought, you know, people wouldn't care about music, classical music especially, but it sort of worked out. And I have a book chapter with Cambridge Press contract, so I'm working on that. So again, when you challenge yourself and try things that you wouldn't expect, I've never had expectation of something like this, you will find doors. And doors will open for you, doors that didn't know exist. Okay, why is there duplication here? I don't want to talk about this, very research focused. I'm going to stop here. There's a little bit of research stuff. I'm not going to talk too much, maybe a little bit, maybe just 2 or 3 minutes, but I'm going to open the floor again to questions, ideally over mic if you like, and then we'll continue on. Sounds good? Okay, any questions, queries, concerns, criticism? C-sounding words.
Hi, Saman. Can you hear me?
Yes, I can hear you.
I don't know how to start because I think your talk was like widely comprehensive. I guess to start, I would say that I felt this tremendous connection to your talk because there are a lot of like, it's scary how similar I think our backgrounds are. I'm also a Vancouverite, and I have a bit of an eclectic set of majors. I'm studying MCB and astrophysics, and I have a tremendous interest in literature and music. And I'll be honest, I took the liberty of stalking your piano Instagram a little bit earlier, and I love your Schubert sonata. The phrasing is just beautiful. But I wanted to ask, clearly you know what you're doing. So how do you have that much time to practice? How do you balance that with your medical school? Because that's you know, both are tremendous time commitments.
For sure. Thank you, Spencer, for the question. It's a pleasure to meet you. It's very kind words, but again, I think the best representation I have of myself is a student, and I think that word is the greatest summary of myself and Maaz, because we're constantly learning, and again, we're humbled by the remarks, but we're still students. In terms of that, how do I do it? I think just getting used to doing a lot of workload. I mean, I was involved in a lot of things early on at Berkeley against people's advice of, you know, don't do this, just do this. You sort of build this, I suppose it's an aptitude, I can't say if it is, but it's something that anyone can build. You learn to work well under high pressure, right? And one of the nice things about all-nighters and cramming things is that our brains become very wired to make as many sort of efficient steps to achieving something. And if you sort of master that with that, I mean, fear is a great motivator. That's probably one of the takeaways of things. Just being fearful of things potentially collapsing helps you stay alert with all the other rushes and neurochemicals. The way I do it is that I sort of craft some time for myself, and that might be at 2 in the morning. I have a digital piano that I can put headphones on. That could be between lectures. That could be as a break in between things. It's just music has been something I really loved. And, you know, in fact, maybe since you brought this up, during my 5 years at Berkeley, even though I did minor in music, I couldn't practice. And I was really sad because to practice, you need 2 or 3 hours constantly with teachers and professors. I didn't have the time or the money. I couldn't do that. So there was no Schubert going on really. But when I came to Toronto, and again, this is before, I'm going on a tangent, but because you asked the question, I want to make it personable. Before I could find an apartment to rent, this grand piano that you see behind me, I went to a different city to rent it out. So I think to answer the question that you have, and this is an overarching theme for everyone, if there are things in your life that you care about, and this could be a significant other, for example, this could really be anything, you need to make it work because that thing is important. That becomes your center and your world will adapt, as opposed to how do you put piano into medicine? Piano is central, you build it around it. And you will find ways of doing it. I'm happy to chat more, maybe if you like, about that later. I hope that addresses the question, hopefully with some other ancillary takeaways, but okay, good, yeah. Any other questions? Pleasure, pleasure. I haven't bored you too much, hopefully, I assume. No, I can't tell people's reactions, so I'll continue, but not begin. No more questions, actually? Richard, are we good with the chat? Excellent, so let me also, 28 minutes.
We are good for the chat.
Great, I'll make it quick. So I do a bunch of things now. With the whole COVID situation, I moved to Montreal about 14 months ago, and I couldn't work in a basic science lab. In fact, I haven't worked in a basic science lab in about 3 or so years. So Maaz is the real scientist here. But I've been doing some projects, one with the Neurosurgical Simulation Artificial Intelligence Learning Center. That was said very fast. But essentially, this is, let me actually see how photos yet. So for example, there are devices that assess your acceleration, your coordination of your hands, how much force you apply. And the goal here, this is done by a brilliant man, Rolando Del Maestro. I have a great respect for him. He's a wonderful man, truly brilliant. And what he's trying to do is to find expertise in something and to develop expertise. You know, you have different strata of students, medical students, residents, staff, and they have all of these interesting statistics that they do to make surgery more safe and beneficial. So that's something I do. I also work in the Translational Neuroimaging Laboratory. Why did I join that? Because I don't know statistics, and the best way to learn something is to get involved in doing it. It's called kinesthetic form of learning. It's very effective. And so I've been working on some projects. These are unpublished works, so maybe this stuff shouldn't be used. Essentially, what we're doing in this study, it's being submitted soon. We're looking at, so we have a PET ligand, and the way that Alzheimer works is that it has a progressive onset, which means it usually starts from the medial temporal lobe, and it goes around the cortices. And these are called the Brock stages, named after a pathologist in Germany who's brilliant because they use the same system in Parkinson's as well. So what you can probably see here are, this is an older marker, this is a newer one, and there are differences based on the groups. So amyloid and tau are the two main risk factors. And we're sort of looking at people that were not cognitively impaired, so cognitively unimpaired individuals, because the whole point of the study is to try to predict when and how there are metabolic changes in the brain, the overall structure, that we can predict the onset of Alzheimer's. Okay, so that's a little bit of this stuff. Again, don't want to make it complicated, and there's no reason to. And you can see different regions of the medial temporal ventral cortex. And again, this is a marker that they use in different gradients of activity. Memory composite z-score is one of the measures that you would sort of operationalize this and see how much memory has declined. I should have also mentioned this, I didn't. In Alzheimer's disease, learning and memory are the two main areas that are affected. So most of the development that is done focuses on those areas, okay? This is unpublished work, so maybe you can take it out. Some work you see.
Saman?
Yes.
There's a question in the chat. What were some significant life-changing books that you've read?
Oh my God. There's a whole list I will give you. They're all life-changing. I'll talk about that afterwards. It's a beautiful question. Actually, let me skip this and just get to the question because I don't want to. Okay, so UC said, forget this. Some surgery stuff we're doing, looking at seizure outcomes and pediatric epilepsy. One of the questions that are remaining is that if someone goes through epilepsy surgery, which removes part of your brain, does that change your cognitive capacity? And FSIQ is one of the measures of doing that. Don't want to talk about it again. It's meta-analysis. This is an important slide. I'm going to stop there. I'm going to answer that question. Could the person who asked this clarify what genre they're interested in? Because I can give you a lot of answers based on that. Because I can recommend books, but I want to make sure that they're in the appropriate genre. And also, I don't know anything about finance, management, and self-help books. So these are, and cooking. So those four things I don't know anything about. Other things I can happily entertain. Could the person clarify the question? Okay, the thing that shaped my perspective, I think one of the greatest books on life. This is a really dense book. I'm still reading it. I was obsessed with it. I even wanted to learn German to read it. I'm thinking of that. I need to learn French first because I'm in Montreal. This is a book written by the Austrian philosopher Ludwig Wittgenstein. and it's called Philosophical Investigations. In German is Philosophischen Untersuchungen. And it's brilliant because the way that he writes is that these are these little sentences and they're aphoristic. So, you know, you read it and you're like, well, what's really happening? This is a really good book because it changed the way that I thought about the mind. It changed the way that I thought about language. The basic premise is that, I don't want to talk about philosophy, I always get there. The basic premise is that the way that we derive meaning is how something is used. So the usage of a word determines its meaning. It's very simplified. It's a lot more complicated, but that's one book. Books that are probably easier to read that you would enjoy. I talk about death. I mean, I'm interested in neurosurgery, so I'm definitely going to mention Paul Kalani's book, When Breath Becomes Air. That's a popular one. I recommend. It's a memoir. You can read it in one day. I did it in one day. It was great. Read it again and cried. Read it again, cried. So recommend that. I'm probably going to curate a list of things and share it with Richard with a brief commentary, so I can give you 5 to 10 options, because that's something I'd be happy to do in different genres. But the biggest thing, though, I would say is books that challenge you. Like if you get stuck with something and you try and you can't read it, those are the best books. That I can tell you for sure, because the more that you try, the more refined you become, right? So it's sort of going against the whole utility of, well, let's read all of the books and all the pages of everything, right? It's like the quality of it and the touch of it. So that's one thing there. I hope that I'm answering these questions, hopefully not in a very centric way. No worries, great. I'm going to end this with the good life. I did this thing on LinkedIn back in 2020. I had this 20 lessons for 2020, and I shared it, and I will read it to you. I think they're somewhat significant. Some of it I did curate, so I didn't write all of it. I modified a few, maybe 4 or 5. Know your why. It's the most essential question that we progressively develop. What do I want? Who am I? Who do I want to be with? What would those changes be when I am with the people that I am? And what does it mean to me? Very important. Second, experience silence. We're so busy with all the things going on, social media, news, all that. Experience silence. Recommend it to everyone. Read, write, and think more, probably in that order. Visit libraries. I'm biased. I have a disease for books, but even when you go to libraries and you see, I mean, by the way, I have not read all of these books. I'm not going to pretend it's a 30-year project for me. When you go to libraries and you see all of this interesting stuff that you don't know, you exercise humility, because that is a great place to feel human. Okay, listen to classical music, highly recommended. Challenge your biases, we went over that. Reverse engineer your goals. You want something, you have to go backwards. This is maybe there's more engineering people here that can probably do a better job than me, but you have to step backward thinking as opposed to forward thinking. We do mostly forward thinking, prospective, but this is a great thing to do. Obsessively focus on overarching goals. If you're serious about something and you love it, you must go after it. And you will find the time to do that. Make your mistakes quickly, right? It sort of relates to also taking risks, right? I talked about that, how and why. Graciously accept mistakes. Accept it, acknowledge it when you're wrong, and say it out, always. Again, in any discipline, in anything in your life, do it. Learn to learn rapidly. Learnability has been related to intelligence. How well you learn something, challenge yourself, find things that you've never read. Read something by Chomsky in linguistics. Doesn't hurt. Constantly synthesize new ideas. Some of those ideas will be nice. If you're working in a Nobel Laureate lab, like Maaz was, 90% of what he did didn't work. Because if you want to have the audacity to do something that's uncharted, you will fail, but that little bit that works is worth it. So it's also risk ratio benefit. Work much harder than expected or asked. You're doing it for yourself. If you love something and you do it for yourself, you're not doing it for your boss, for your PI, whatever, you're going to do it better. Find excellent mentors. At every stage of our lives, there are people that have wisdom, that have knowledge, that have a background that can help shape us. Seek them. I sought them. I continue to. If I can be a mentor to any of you, I'd be delighted. Collaborate with intelligent and motivated colleagues. That's self-explanatory, and that's probably easy to do at Berkeley and other places too. Maintain passion and persistence in all endeavors. That's just my personality, so that's what I would say. Trace your roots. Understand what has gone before you, because I mean, on two levels. One could be like a sociocultural analysis I don't want to get into. The second is that when you think about an idea, right, that idea was modified many, many times until we got to that stage. And that's extremely important, right? People would say, well, you know, the heliocentric model was terrible because, well, it sort of flawed us. No, knowledge, the way that knowledge builds is through errors. And those errors reform the way that we understand things. So I would say the work of Plotinus is as important as Copernicus. Don't quote me on that, though. Learn how to say no. I don't know how to do that in many areas, and that's something I'm working on. Not all questions are worth pursuing. Not all thoughts are worth entertaining. I'll leave it open. And the last one is death is closer than we can imagine. Thank you. That was my presentation, I think. Follow me. I mean, you're not in McGill, but if you're there, I'm doing a TEDx talk actually coming up. So if you're interested, it's on music and medicine and philosophy. I'll share it with you guys. This is my platform. I'm available to answer questions. I may not be able to do it immediately. Any help that I can give, I will be more than happy to. I think I went a little bit over time. I apologize, but I hope that this was helpful to you. And I'm going to check the time, 39 minutes, 10 minutes over time. Not that bad. Okay. Questions, concerns?
I'd just like to point out, Saman and I, we've been talking about a project, like a mentorship project. So we're going to be compiling data together of like all the successful people who've kind of gotten into different programs or like different places. And we're happy to mentor you, speak about like different aspects of like grad school life and stuff. So please feel free to reach out to us on Twitter or Instagram or even LinkedIn, whatever's easy for you.
And if we don't know the answer to your questions, we will redirect you to people that do. So we'd be delighted to do that. Any questions? And I also will get, I think I read all the chat questions. So yeah, I'll give you a list of book recommendations too.
Yeah. Just as a note, we will be including all of the post-presentation materials on the Notion calendar, which is available on our website. So basically, if you see the entry for today's meeting on our website, you'll be able to click on it and it'll have a compilation of all of our resources post this meeting, including access to the recording without information about the unpublished research. So, but yeah, does anyone else have any questions?
I have one just about like general McGill culture for medical school. How is the culture in terms of being able to explore different avenues? It seems like both of you guys have been able to do that, but I was curious if that was something that was encouraged by the general population at McGill, or if that was just something that you guys were intrinsically went to McGill with, and it's an anomaly.
Thank you. Maybe the McGill part can also be taken out just for the sake of it. Richard, I'll happily answer the question. I think my interests are a bit of an anomaly because as you can probably see, I mean, I'm interested in a lot of areas of medicine too, but the nice part about McGill is that if you want to do something, there's never someone saying no. This is probably true of Berkeley too. I mean, I remember one of the things back in the day when I was trying to like go over the unit cap, they're like, you know, you can't do this, you're going to mess up your GPA, or, you know, you can only do like 2 majors, 1 minor, whatever. You make it work. And I think that if you're with supportive people, or if you know roughly why you're doing what you're doing and you see value in it, then it's certainly possible. I mean, most people at McGill, I mean, I can't comment on other schools. There's a lot of interest in public health and social equity and other things, as is in Berkeley. That's something I'm not particularly gifted in or have as much talent in. So I know that I wouldn't be as effective in there, for example. So I do more research sort of things or academic things, but there's a high degree of flexibility at McGill. I don't know much about NYU. I'm sure it's the same, but I'll pass it to Maaz to comment on that.
Yeah, I wasn't sure if this was a McGill specific question or not, but I think cultures, scientific culture or like culture in general can be really different. So right now I'm going through this phase where like it's really hard to adapt, especially after leaving California, kind of adapting to how fast things are in New York. So it's really about like finding the right network. So I'm looking for mentors right now. So I'm actually in the same phase as probably some of you are, like some of you might be seeking mentors. So I'm like, you know, you have to forget the past and like start from scratch and like seek mentors and kind of develop a network that's going to be helpful to you. So I think that's what makes you feel more comfortable in an environment. Obviously, the way people do things is going to be different. And I think that's not something that's important. What's important is that no matter how they do things, you have people around you that can support you. So I think one of my most important experiences at Berkeley was that whenever I wanted to try something, there was someone who was willing to, you know, go all the way with me, especially when I was doing the bioprinting project. There was no lab that was willing to take me up. I didn't have lab space. So I had to really struggle with this. We had people who were willing to kind of not do internships and work all summer on this project, like in like nowhere, basically, you know, like, so it's really about finding the people who will like fight with you, be with you at all times.
Does that hopefully answer your question, Sophie, or we good?
Yeah, it does. I was just curious because culturally speaking, my family's French. So I was looking at McGill as like a possible school. And I know for undergrad, it was a little different. Yeah. So yeah, that's why I
don't have
asked. Thank you for the
background,
insight on NYU. Yeah, if you ever want to practice, let me know. but yeah.
Well, I got to start
Thank
from
you for
zero,
answering.
but I will.
Yeah.
Again,
Anyways, thank you, Richard. I'm gonna have to log out now. I have another meeting soon. So anyways, thank you everyone for being here and kind of listening to us. And Saman, always nice to kind of link up back with you.
Likewise. Thank you. I can stay for another 5 minutes or so, but if there's no questions, I'll just stick around then leave. So yeah. And thank you for coming. And thank you, Richard, for organizing it.
Thank you. Have a good night. Emil, you have any questions? Nope.
Yeah.
No, guess not.
Okay, no worries. I know some people are shy, but I guess I went over the time. So anyways, people are busy too. Good. Thank you again. And